Welcome, everyone. Thank you for joining us today. We're going to be giving you kind of a whirlwind tour of CartograPlant over the next about 2.5 hours. And so I'm going to start today by just giving you an introduction as to what CartograPlant is. Then we're going to move into an introduction on submitting data to CartograPlant, and Emily is going to lead that. And then from there, we're going to start to talk a little bit about data collection and mobile phenotyping. We're going to be focusing on the application TreeSnap, with just a couple notes on a new application that will be coming up in the future, WildType. And Abdullah and Ellen will be talking to us a little bit about that. After that, we'll take a short break, and then we'll return to talk a little bit about behind the scenes of CartograPlant in terms of environmental layers and data. And then we'll get into the weeds in terms of analysis with CartograPlant, so talking about GWAS and landscape genomics. And so Irene and myself will talk to you guys a little bit about that. And let me just go back here. And then we'll wrap up with any questions that you guys have. So for today, please feel free to send questions into the chat as we're going along. We're going to try to address those the best we can. I'm also recording these sessions, and I'll be sharing that with you after the close of the workshop as well. So if you don't get all of that material, you'll have an opportunity to look back over that as well. And before we get started, I just want everyone to go ahead and sign in. This link is here, but it's also in the email that she sent right before the workshop. So just take a moment to let us know where you were from and who's here, and if you'd like to hear more about CartograPlant after this session as well. So in terms of how we're going to structure today, we're going to be, like I said, kind of starting with background. We're going to be demonstrating what CartograPlant can do. We're going to be highlighting several of the features. And if it's something you'd like to interact with, you just have to apply for an account through TreeGenesDB. And you'll see in the link that she sent out, there's actually a link to a handy guide to get started with CartograPlant, and this will walk you through how to get that account. And we'll try to approve those accounts as those are coming in too, so that you can either submit data or start to analyze data with your account. If you're looking to kind of browse and play with CartograPlant, that is the URL that's available there. I've also put in the link to the TreeSnap mobile application. This is predominantly tree specific. The WildType application that we'll be touching on as well is broader than just trees. And I know everybody who's attending this is coming from, some of you work on trees, some of you work on other species. But if you'd like to follow along there, you can download the TreeSnap application, mobile application there. And then again, from that link that Shay provided, we have all sort of the Git links available for you so that you can follow along, and also the agenda there. So just keep all of that handy and go back to those resources as we move along. So just to give you a little bit of background about what CartograPlant is, this is essentially a project that has been focused on developing a user-friendly and map-based platform to facilitate meta-analysis across scales. So truly this idea of a genes to environment perspective. And so who are our target audiences for CartograPlant? So it can be, focused on conservation genetics research, looking at invasive species, looking at pests and pathogens. This also is an application that appeals to the plant breeding community, especially in terms of identifying resistant and new cultivars for crop species, also in terms of breeding existing cultivars. And this also is an application that's been used by the evolutionary biology community in terms of understanding how plants are evolving and adapting to a changing climate. And so these are just a few of the applications that CartograPlant is useful for. It also really represents the type of studies that we curate and target to put into CartograPlant, and we'll be talking a lot more about that. And so CartograPlant was actually originally born out of the TreeGenes database. It is now, as part of our USDA funding, has been expanded to go beyond just trees and also encompass all plants, especially plant studies that have georeference accessions associated with them. But when we started, this application was actually called CartograTree, and it was focused on tree species, and it was connected to our tree genes database, which is maintained today. And this database holds over 2,000 species at this point and holds a variety of genetic and genomic data associated with them. So genomes, genes, genetic maps, protein sequences, markers, and also phenotypes. And so this collection of data was really the basis for developing this application. And this application, as well as several databases, are actually hosted by Triple. And this is an open source framework that it's developed in order to basically simplify bringing genetic, genomic, phenomic content online to independent research communities. And so Triple has been in development for several years. There's now been a recent release of a new version of the Triple framework. And this, you can actually download, interact, set up your own instance of Triple by going to triple.info, which is the homepage that I'm showing here. And you can also get an idea of the other databases that are using Triple around the world. And so it's actually used by, at this point, over 40 different databases spanning the globe. And a lot of these websites are actually very focused around plants. We have a few animal examples in there as well. But you can really see the diversity of applications here: rice, cotton, horticultural tree species, citrus, legumes, basically a really wide range of plants and crops that are represented in Triple. And all of these databases were able to be developed using this basic framework because it has within it the ability to store genetic, genomic, and phenotypic information and allow these independent research groups to get off the ground running and sharing this information with their communities. And so TreeGenes is one of the databases that uses, just one of the many databases that uses Triple, and CartograPlant is one of the applications that's developed from that. And so just to kind of give a quick view of what we mean when we say Triple, we think of Triple as essentially a framework, and that framework has a web front end that can be customized, and that's using a content management system known as Drupal. And then it also has a back end to it, and this is the database. And this currently interacts heavily with the Chato schema, which is developed through the Gmod project. But the current implementation and future implementations of Triple are far more flexible than that, and will be able to work with a wide range of data organizations. But essentially that front end and that back end provide a framework that's already ready to go for a lot of the standard things that you're going to do with a genetic, genomic, or a breeding database. So things like how to hold genes and markers and QTLs and serve those out to the community. In the more recent releases of Triple, this also includes the ability to perform analysis, and that is what paired with the Galaxy framework. And many of you may have heard about Galaxy as an open source framework that is an independent project that allows users to interact with command line bioinformatics software and run analyses in real time. There's a public instance of Galaxy that's hosted, but there's also the ability for individual groups to host their own instances of Galaxy. And that's what we're doing here. And we interact with this within the Triple framework for our application. One of the primary advantages of working with a shared framework for developing databases and applications built on those databases is that we can share specific applications with the community and develop those around standards that make those applications easy for other groups to implement into their databases. So specific crop groups or specific research groups may have an interest in having certain functionality, and they can create a module within the Triple framework and then share that module, and others can implement that in their system. So Triple kind of provides that basic framework that with the features that are shared by many databases, and then allows that flexibility and community environment to develop other applications. And so what you'll see here is I'm showing that based upon this Triple framework, we're going to be talking about two of our applications today: CartograPlant, which plugs into this system, as well as the Plant PopGen submit pipeline, which is how we get data into CartograPlant and into the database. And so that's where we'll be focusing today. And so if you go over to CartograPlant.org, that's really where we're going to be focusing, talking about today. This is, as I mentioned, is now representing a wide range of species. And if you head over to our landing page, you'll see information on getting started, which will bring you right into the web-based framework. You'll also see a link there that says submit, and that's going to let you go in there and actually start to submit data to us through that tips module I mentioned. And then there's also a user guide there to give you some background on how to interact with CartograPlant as well. And so that's what you'll find when you come to our homepage. So if you hit that Get Started button, you will end up on the interface for CartograPlant. And so that's going to give you a view of the whole map, all of the trees that are available through CartograPlant, and a summary over here under the map summary of what's currently available in the framework. So you can see the total number of accessions, you can see the number of species represented, the number of publications, and also if we start to select some environmental layers, you'll see that information updated here. But just to pull back from a moment, what is CartograPlant? We're essentially trying to integrate genetic and phenotypic data for forest trees and other plants with environmental data through a map-based visualization platform. And so the key there being that we're trying to enable visualization of both the map, but also of the data, advanced search capabilities, and we're trying to do this in a way that's biologically relevant. And so you'll see examples of that as we move through today. All right, so here's a view of our homepage. And what I want to highlight here is that we've selected and turned on our environmental layers tab. And what you see appear when we do that is a whole set of layers. And I think we have over 900 environmental layers that are available now. And these layers encompass both what we would call regional layers, meaning maybe things that are specific to specific countries, continents, but also the vast majority of these are actually global layers. So these are available for most of our georeferenced coordinates around the globe. Oops, let me go back over there. And so here is just a zoomed in view of our environmental layers. So one of the key aspects of working with CartograPlant is that the metrics are preloaded for the georeferenced coordinates for our plants, and they're available but they're also available for all locations on the map. So if you are clicking around on the map and you have specific layers loaded, you'll actually get the metrics associated with that layer at any location, but we'll specifically hold and optimize the metrics for the georeferenced plants as the studies are brought in, and that's going to enable us to perform analyses downstream. You're going to hear a lot more about the layers from Rish, so I'm going to switch next and talk a little bit about and give you some examples of what these layers look like. So in this example, once we have our environmental layers tab open, we can actually scroll through these and expand all those categories that you saw on the previous slide. And for example, if we want to select our NDVI layer, we can turn on that layer and that will load on the map. Once we have that turned on, we can then use this info button to get more information on that specific layer. And so there you will find the citation from the origin of that layer, description of what that is, the resolution of that layer, and then also information on basically when that information was last updated, any relevant units, and so on. One of the features that we specifically have for the layer is the ability to adjust the opacity of that layer. So if you're playing with multiple layers on the platform, you have the ability to kind of layer them in different ways and modify that view here. And in this case, I'm going to zoom in here to another example. So here's our canopy height in 2019. And so the same applies here. If you were to collect or set to any of these info buttons, you would get information on this particular layer, where it came from, how it's calculated. And then if you hit this legend key here, you will see the legend for a given layer, what the units are associated with that layer, what it's measuring. So in this case, again, canopy height, this is set to meters, and the darker the color, the higher the canopy height is. And I've zoomed in here on South America, so you can see an example of that gradient on the map. And we've got our opacity turned all the way up so we can see that very well on the map as well. And so, like I said, you can turn on multiple layers at the same time. You can you can layer those layers in different ways. You can get information about what's there, and just sort of scroll through and explore what's available through the pipeline. So I wanted to show an example of some of our more heavily used layers. So WorldClim is obviously widely used for analysis. So here's an example of a precipitation, one of the precipitation layers in WorldClim for the month of January. We have this layer turned on here, and we have essentially our legend available here. In a lot of cases, we actually modify these legends so that the data is a little bit more viewable to the users. And so this information is here. The units are listed as being in millimeters. We've turned that layer all the way up so it's nice and visible. We've zoomed in over here to Bergen, Norway, which is known to get a lot of rain. And so you can see that visualization here as well. I wanted to also demonstrate the fact that you can select a point on the map, as I mentioned. So you can click anywhere on this map with the layers that you have loaded. And here you can see that you get a precipitation value of 226 millimeters based on that location. So you can click anywhere. That's not where a tree is currently located from a given study or a plant, as you can see, but you can still get that information here. And if you were to click on the specific plant accession, you would get both the details of the plant itself as well as the environmental information. And another important note is that if you have selected, say, multiple layers that are up, say you've got your precipitation, you've got temperature, you've got NDVI loaded, you've got canopy height loaded, you would actually get the variables and the information for all of those layers at the same time in that view. And we always have the elevation and lat long at the top of that box as well. All right, so here's another view looking at major soil types. This is another layer that's widely used by the community for a lot of different types of analyses. And you can see, just like the others, we have the ability to turn that layer on. We can get information about that layer. We can adjust how that layer is visualized. And all of the colors that you see are defined according to specific soil types. And so if you're performing an analysis based on soil type, you would be able to collect all of the metrics on soil type associated with the georeferenced coordinates of the plants you're interested with and bring that in for analysis downstream. And I also wanted to highlight our range maps that are also available in CartograPlant. Now, right now, most of our range maps are focused more on tree species, but we'd like to start bringing in more for other plants as well. And so we definitely welcome, and I'll be talking about ways to talk to us about different layers that you want to see available in the CartograPlant interface for your own analysis as well. But I wanted to specifically highlight when we look at layers that are associated with basically a single value, maybe you're looking at a range or you're looking at something that's just going to give you kind of a view of how much is covered by a specific layer, you have the ability to actually select the color for that layer and then adjust, again, the opacity and sort of look at overlaps of ranges or select plants that might be overlapping ranges and so on. And so you have the ability to sort of customize that view and then select as you move forward there. And just a note here on this side, as we are turning sort of on and off layers, we're not doing anything with our plant selection at this time, but you can see that you can see how many of those layers you have loaded, which is helpful because this is a long list and you might have lots of things turned on and you can kind of view what you're looking at any given time. And so over here, I also wanted to highlight one of the newer features that we're putting together. This one is not fully implemented yet. But one of our goals is specifically to be able to load up the population structure layer for any of the studies that we have available that are compatible with that. And so we reference our studies with these TGDR accession numbers, which we'll be hearing a little bit more about. And for this particular layer that we've turned on, you can kind of see these dots here, and these represent plants that are part of this study. And the gradient that you see there actually represents the genotypes. And so we'll have that kind of pre-calculated on a per study basis with the ability to load just the population structure overlay layer for a given study and visualize that on the map. Now, this will sort of interact with our workflows, where if you're performing a meta-analysis across 2 studies, you'll have the ability to recalculate that population structure based on the integration of both of those studies and then generate that layer on the map. So we just have that example loaded now, but we're moving towards making that available as well. So essentially, our environmental layer goals are to integrate environmental layers from curated public sources. So right now, we're very focused on precipitation, climate, land use, human impact. We've got fire, soil, elevation, slope, and a variety of plant inventories. Like I said, let us know what you'd like to see in this framework. We're constantly updating these layers as new information becomes available, and we do a lot of work to make sure these layers can interact with one another, which is a challenge in and of itself, which we'll talk a little bit about. And then we also have our range maps. And like I said, these are primarily focused more on our tree species right now, but we'd like to expand this to a lot of other plant species as well. So definitely come talk to us there. And right now we're primarily working with layers that have been extracted from LiDAR applications, but we also have a couple of current collaborations where we're working with how to kind of get LiDAR related data directly into CartograPlant. So we'd love to talk to people who are interested in doing this as well. So again, tell us what layers that you're interested in seeing in CartograPlant. So now I want to move away from the layers and talk a little bit about where the plants are coming from. And so our goal with this application was really to provide a mechanism for georeferenced plant data to be imported directly from scientists. So this is the most ideal location, my most ideal workflow for us to obtain our data is that at the time of publication, you're ready to publish your paper, you come in here and you submit your data, you know the most about your study, so you can give us all of that information. You get a long-term DOI, your study appears on that map, and it is then available for you and others moving into the future, especially as a mechanism to build upon. One of the things that you'll hear us talk a little bit about is that our goal is to use all of this information if it's properly tagged in order to give it value moving into the future. So if your study, for example, didn't have a reference genome to start with, do we have enough information to map those markers onto a reference genome when it becomes available? Or if that study worked from an older version of a genome, can we map those markers onto the current version of the genome so that meta-analysis can be performed across multiple studies? And so we ask a lot of questions when we take in this data for the purpose of making sure that that data is represented accurately and also that it can be used into the future. The second way that we're moving data into this framework is through mobile applications. And so these are both citizen science and research-based applications. So things like TreeSnap, things like WildType, you're going to hear about those today. And we're already bringing that data into CartograPlant and making that available to the users. One of the nice things about the mobile applications is that they are often specifically targeting phenotypes. And those phenotypes are specific questions that either citizen scientists or landowners or researchers are answering and providing information. And this can be particularly helpful, especially for pests and pathogens. And so you'll see some examples of that today as well. And then the last way that we bring in a lot of this data into CartograPlant is through public repositories. And this is sort of the harder way to do it. We actually have a team of biocurators, our undergraduate team who works very hard to do this, but they often work from Dryad or they work from other public repositories to try to format the data, fill in the blanks of how data should be represented, and then get it in there. You're going to hear from Emily in a little bit about how hard that is, and some examples of, getting data into the format that works for us versus kind of us trying to figure out all the unknowns from working with these data sets. So we'll talk a little bit about that as well. So again, direct submissions from scientists, and we're getting these we run our public competitions, we work with journals to require data submissions, and we provide a free DOI at the time of submission for anybody who's getting that information into us. We also, again, as I mentioned, are getting our studies curated by our curation team. And then we're also pulling additional trait data from public sources as well. So BN being one of our primary sources, and then also TreeSnap. So this is, again, our citizen science landowners and researchers using this application to phenotype their populations. And we'll talk briefly about wild type as well. So on the analytical side of CartograPlant, one of our primary goals is to be able to enable our users to perform landscape genomics, look at population structure, perform association mapping, so genotype to phenotype, obtain diversity estimates. So basically have this data labeled across studies in such a way that we can enable new analyses based on this data and meta-analysis across multiple studies. And so that's where getting these studies into the framework or into the CartograPlant interface in a way that's well accurate will really enable this much more. So here's just a few screens of where we head. So Dryad's one of our primary places for our biocurators to pull a lot of the information from these studies. This can go really well, or this can be really hard. So we'll talk about that. And then that information actually gets brought in through this CartograPlant PopGen submit pipeline. So this is actually an independent module that brings data into our database and then serves it back, and then we serve it back out through CartograPlant to our users. And so this is where we actually have two sides to this. We have one side that the public interacts with, and then we have one side that our biocurators interact with. And we ask a lot of questions in order to bring all that data in, and Emily will be talking more about that. And then you'll also hear about our mobile applications, so primarily TreeSnap today from Abdullah, and how we bring that information into CartograPlant. And I'll touch on BN a little bit in terms of bringing in trait data. And then finally, what I want to just show here is we've got a kind of a zoomed view of our CartograPlant application. And one of the things that I want to show here with the data is that, and actually let me just go back one screen. Nope, that's the right spot. If you select a given accession on the map, any plant that you're looking at, you're going to be able to obtain environmental data if we have any of those layers loaded. And you're also going to be able to view the entire information on the study. So in this case, you're first looking at the individual plant, in this case, selected here. And what you actually see is that there's several individuals set at the same location. So this could be an example of, say, a common garden at a specific site where we have all of the trees sort of set to the same georeferenced location, but potentially maybe some of the source materials coming from other locations, and that can be captured through CartograPlant as well. And so if you were selecting a study in this case where everything's sort of grouped together in one location, you would have the ability to select all the trees and move forward with an analysis, or you could go over here and just select specific plants that you want to work with and move forward with the analysis. And what I want to sort of highlight over here on this other side is our ability to search this information. So you can search by things like your study accession number, if you know that. You can search by the title of the paper because you're more likely to know that. You can search by species information, by phenotypes, by marker type. And so these are all the filters that you can work with on this side to display the information over here. So once it's displayed, you can also view, I was going to show the study information, but we'll hop over here first. I'm going to show another study here. You can also search, as I mentioned, by different phenotypes. Emily's going to talk a little bit about the role of ontologies and sort of getting us to a point where we know that multiple studies have worked with the same plant structure or have measured the same phenotype, which of course everybody calls things, labels things a little bit differently in their publication, but by kind of standardizing that on our side, we're able to provide searches where you can see all of the studies that maybe have measured this particular trait. And so here's an example of where we're searching plant structure equal to floral organ. We zoom in on a particular study that's done this. And you're able, again, to select, you can zoom all the way in on that study. You could select all the trees from that study, or just look at individuals and select individuals for downstream analysis. Now here, Again, you can, this study is an example where you do have different georeference coordinates for each of the plants in that study, and you can kind of see them spread out here on the map. And so you would have the ability to just grab all of them without clicking on them each individually or doing it that way. And so another view that I want to show here is the ability that if you select that show study information button, that you'll be able to go in and get a detailed study view. It will tell you what was included in that study type. So in this case, it's a genotype by phenotype study. If we had independently measured environmental variables, we might say genotype, phenotype, environment. We know that we're working with microsatellite markers. We know how many markers we have and how many phenotypic measures we have. We're able to download those files ourselves if we want to just take them and move them onto our local computer. We can download them that way, but we can also select this entire study and move into analysis framework from here. Here's another example of being able to search information sort of at the study level. So I mentioned being able to search by study accession here, and you've got an example again of a study with multiple georeference points on the map. You can select that entire study or individual plants and move forward from there. And here's an example of being able to search by the species level. So say you actually wanted to see all of the studies or all of the accessions related to a given species. So here we're selecting sugar maple, and you're able to see all of the study, it could be coming from a lot of different sources in terms of study information, or one of our mobile applications. And we actually have a mix of things being shown here. So we use our purple icons here to indicate that this is coming from one of our mobile applications. We use sort of our green icons to indicate that it is coming from one of our curated studies that are coming in either through the user or through our biocuration team. And you can actually see that on the map as you're kind of zooming in and out to see where the source of that material is. And right above this, which I haven't talked about yet, you can actually turn on and off the different plant layers. So if you just want to view things from TreeSnap, you can do that. If you just want to view things through Dryad, you can do that. Or through kind of our direct submission, which is labeled as tree genes, but isn't necessarily a tree. So you can turn on and off all of those layers to see what's available. And as a note, I wanted to also show what this sort of looks like after a study gets submitted, because another way to view this is if you've submitted your data or you're looking up another study that's been submitted, is that you can come in here and actually view the study details for that study. And you can link out to CartograPlant here and then actually view the study there and start to interact with it. And here is also another place where you can download those files and find out even more about that, the background of that study, link to the publication, and so on. We have this little Google Maps view when you're looking at it from this perspective, and that's more just to kind of give you a high-level view of how many plants are involved and generally where they're located. It also helps the user when they're submitting their data to make sure their georeference coordinates are in the right location. So that's just your quick view, but if you hit that view in CartograPlant link, it's going to bring you out to the study in the CartograPlant framework. And here you see, we've loaded up that particular study accession. You see the same species that we were looking at before, and you can view detailed information from that study. And if we look at a detailed view of that study, just like before, you can also see, again, link out to our study information. This time we have SNP markers rather than microsats. We have the total number of markers that are involved in this study. And that's all information that can now be moved forward with for analysis. And just a quick note, you also have the ability to not only just search things by species, by phenotype, by study ID or by title of that study. You also can just come in here and put a square. It's kind of hard to see this, but you can put a square around a group of trees that you're interested in. You can form a polygon and actually just push those into analysis as well. So say there's just a subset of individuals you want to work with, that's a functionality that CartograPlant supports as well. And that's where that is. So we've talked a little bit about in the environmental layers, kind of the scope of what we currently have and the ability to upload more of those layers. We've also talked about where we're getting our plant information in terms of what studies go in and how we curate those. And I should say we're open to studies from all georeferenced plant studies and also a lot of different designs. We can host things like growth chambers, common gardens. So a lot of different designs are actually supported through our data import pipeline. And then we also have information about the analytics. And I'll talk a little bit just briefly about that. So once you've selected the plants that you're interested in working with, so whether you've searched by study, a group of studies, by species, by phenotype, those can then go into this analysis workflow. You have to have an account to perform the analysis, so you can save everything as you're moving through. And this is what's essentially going to allow you to filter data, visualize data, and analyze data. And as I mentioned earlier, we do this and we support this analysis through the Galaxy framework. This is an open source, awesome project that exists independently. We're looking at sort of the public view of the Galaxy framework. But essentially the way that we use Galaxy is as an independent instance, so that if you are here, you've logged in, you've hit your analyze button, you've brought your plants in for analysis, you want to grab some environmental data, you want to filter information, everything that's kind of doing the hard work of processing that data and then bringing it back is done over here in Galaxy. And so we'll be talking about some examples of that analysis as we move through. And with that, I'm going to let the next folks give you some more information on what's going on in CartograPlant, but I wanted to first, before we get too far, acknowledge the team. So Irene Cobo-Simon is a postdoc who actually is now working back in Spain, still participating in our work here on CartograPlant, and you'll hear from her today, and she's done a lot of work on the workflow side. Rish is our lead developer who works on the UI aspects and also backend aspects of CartograPlant. Umed, who's a recent addition to our team, has been focused on tips and the tip C component. Emily is a lead developer of TreeGene, so all things database, how that data gets held and how that data is optimized. Sean, who's our lead Galaxy developer. Shay, who's also a recent addition of our team, who's our TreeGenes and CartograPlant coordinator. Oops, there's more. Meg's Stan's group, who includes Abdullah, who's working on both the TreeSnap application and WildType, who you'll be hearing about today. And our curation team leads, Charlie Demerjian, who's just graduated, and then just about to graduate, and then our new team lead, Megan Miles. And our awesome curation team, which you'll hear from Emily Grau how tricky what they do is. So Emily Strickland, Victoria Burton, Maddie Gondonsky, and then also Nick Herndon's lab, who I believe Joe is on the call with us today as well, who's contributed a great deal to some of the visualizations that we're using. And then I also want to acknowledge Ellen Crocker, who you're going to hear from today, who interfaces with a lot of their landowners and people using these applications in different ways. And you'll hear a little bit about that as well. And so with that, I'm going to stop sharing now and we'll move to the next part.